For over a decade, archaeologists stared at an impossible problem. Göbekli Tepe, the world's oldest monumental structure, built by people over 12,000 years ago, held the keys to understanding human civilization itself. But when geneticists tried to extract DNA from the site's remains, they found something shocking. There was almost nothing to analyze. The collagen was gone. The bones had surrendered their secrets to the Turkish heat. What they discovered instead would force them to piece together an entirely hidden history using fragments from nearby graves. And what emerged was a genetic story so profound, it rewrites everything we thought we knew about how modern humans became who we are today. In the early 1990s, when German archaeologist Klaus Schmidt first began excavations at Göbekli Tepe in southeastern Turkey, nobody expected to find anything remotely like what lay beneath that earthen mound. For decades before Schmidt's team arrived, surveyors had noted the site, speculated it was man-made, and moved on. The scientific world wasn't ready for what Göbekli Tepe actually represented. Here was a sanctuary. Here were massive T-shaped limestone pillars carved with meticulous precision, some reaching nearly 20 feet into the air. Here were circles of stone arranged with geometric intention. Here was monumental architecture constructed by people who, according to everything archaeologists had been taught, shouldn't have possessed the organizational capacity, the leisure time, or the motivation to build anything of the sort. These were hunter-gatherers, mobile people following seasonal migrations and animal herds. They didn't build, they didn't settle. They didn't create permanent structures, and yet here it was. What made the discovery even more confounding was the dating. Radiocarbon analysis pushed the earliest construction at Göbekli Tepe back to approximately 9,600 BCE, or roughly 11,500 years before the present day. That meant Göbekli Tepe predated the Egyptian pyramids by almost 7,000 years. It predated Stonehenge by 6,000 years. It predated every other monumental structure on Earth by a staggering margin. Here in the hills of what is now Turkey, at the very tip of the Fertile Crescent, where the Euphrates and Tigris rivers began their ancient journeys, a civilization had gathered to build something that defied everything. Archaeologists thought they understood about human cultural development, but understanding the people who built Göbekli Tepe proved extraordinarily difficult. While the stones themselves offered a wealth of information, while the carved reliefs of animals and abstract symbols told stories about their world, understanding who these people actually were at a biological level remained elusive. That's where genetics was supposed to help. That's where ancient DNA, a field that had revolutionized archaeology and anthropology in recent decades, was supposed to provide breakthrough answers. And yet, when geneticists approached the problem, they encountered a frustrating wall of biological silence. The problem was deceptively simple. DNA degrades. It degrades quickly in hot climates. It degrades in acidic soil. It degrades when bones aren't buried properly, when they're exposed to sunlight, when they're handled repeatedly by excavators. And southeastern Turkey, despite its archaeological richness, is not a forgiving environment for DNA preservation. The high temperatures that characterize the region work like an accelerator on the natural decay of genetic material. Collagen, the protein that binds DNA and helps preserve it, simply breaks down too rapidly to be useful. Researchers at institutions like Hacettepe University explicitly noted that DNA analysis at Göbekli Tepe itself was impossible. They found almost no human remains that were appropriately buried, no skeletons that retained sufficient collagen to yield genetic information. It was a cruel irony. The most important archaeological site in the world when it came to understanding civilization's origins was genetically locked. But here's where the story becomes genuinely interesting. Scientists aren't so easily deterred. If Göbekli Tepe itself wouldn't give up its genetic secrets, perhaps the surrounding region would. Perhaps the people who visited Göbekli Tepe came from settlements where bodies were buried with more care, where bones were preserved in better conditions, where genetic material could still be extracted. This is how researchers came to focus on nearby sites, particularly Çayönü Tepesi, located approximately 300 kilometers away from Göbekli Tepe, in what is now Diyarbakır. Here, at a Neolithic settlement dating to around 8,500 BCE, archaeologists had uncovered numerous burials with remarkably preserved human remains. And from these bones came something that had long been unavailable: actual ancient DNA. The team at Hacettepe University, led by researchers including Professor Yilmaz Selim Erdal and Associate Professor Ezgi Altanışık, began extracting and analyzing the genetic material from these remains. What emerged was nothing short of revolutionary. The genetic structure they uncovered told a story of population origins and migration that fundamentally reframed our understanding of how the first civilizations came to be. The people of Çayönü, and by extension, the people who built Göbekli Tepe and populated the broader region, carried a genetic signature that could be traced to specific haplogroups, specific family lines that had deep roots in the ancient world. The most significant discovery centered on something called haplogroup G, specifically its subclades. To understand what this means, you need to know that haplogroups are essentially genetic family trees. They trace male lineages back through vast stretches of time, identifying which paternal lines exist in modern populations and, by extension, which ancient populations contributed to our current genetic makeup. Haplogroup G is unusual. Its origins trace to the Fertile Crescent, that crescent-shaped region stretching from Iraq and Syria through to western Iran. But more specifically, geneticists have narrowed the origin point further to the northern reaches of that region, to the area where the Euphrates and Tigris rivers begin their descent, to the mountains and plateaus of eastern Anatolia. That's the region where Göbekli Tepe itself is located. Haplogroup G had an unusual history. It emerged and remained relatively isolated for tens of thousands of years, evolving slowly within small population groups. This isolation meant fewer genetic mutations, a more homogeneous genetic structure. The slow evolution of haplogroup G is actually a key marker that geneticists use to trace ancient population movements. When a population evolves slowly, when mutations accumulate gradually, researchers can calculate backwards to determine when specific lineages diverged and when populations separated. The hunters-gatherers who carried haplogroup G were organized into small bands, semi-nomadic groups that moved seasonally, that followed game animals, that exploited wild plant resources. These were the Anatolian hunter-gatherers, and they were among the people who gathered at Göbekli Tepe for ceremonies, for rituals, for purposes that archaeology is still struggling to fully understand. But something extraordinary happened to haplogroup G, something that would eventually reshape the entire European continent. Around the time that agriculture was beginning to emerge in the Fertile Crescent, around 11,500 years ago, the people carrying haplogroup G underwent a profound transformation. They stopped being purely mobile hunter-gatherers. They began cultivating wild wheat and barley. They began herding domesticated sheep and goats. They began settling in permanent villages. And here's the crucial detail. They began expanding. Haplogroup G, which had been relatively geographically confined, started to spread. It spread west into Anatolia. It spread north into the Caucasus Mountains. It spread through the Mediterranean. It crossed into Europe. Over the course of 2,000 to 3,000 years, haplogroup G, the genetic signature of the Göbekli Tepe civilization, dispersed across the continent in what geneticists call endemic diffusion, a wave of advance where farmers expanded into new territories and gradually replaced or assimilated the local hunter-gatherer populations. This expansion was transformative. As haplogroup G farmers moved into Europe, they brought with them not just their genes, but their entire package of innovations. They brought agriculture. They brought animal domestication. They brought new social organizations and religious practices. They brought the Neolithic, the new Stone Age, and they left genetic fingerprints everywhere they went. Today, about 19% of Europeans carry haplogroup G2A, the subline that traces most directly back to the Fertile Crescent. In Sicily, that figure rises to 21%. In parts of the Caucasus, it's even higher. In the Mediterranean region, it's remarkably prevalent. When researchers trace the geographic distribution of haplogroup G2A across the continent, they find a pattern that matches almost perfectly with the archaeological evidence of the Neolithic expansion. The genetic data confirms what the pottery shards and house foundations have been suggesting for decades. Europe was populated by waves of farmers emanating from the Near East. But why should any of this matter? Why should we care about genetic markers that have traveled through thousands of years of human history? The answer is because these genetic signatures connect us directly to Göbekli Tepe. They connect our own biology to that monumental structure built by hunter-gatherers over 11,000 years ago. They suggest that the impulse to build, to ritualize, to create permanent sacred spaces was something that traveled with populations as they dispersed across the world. The genetic story is also a cultural story. It's a narrative about transformation, about how a population adapted to radical changes in their environment and in their society, about how innovations emerged, were adopted, were spread. The genetic evidence also reveals something equally fascinating: diversity within unity. While the primary genetic signature of the Neolithic expansion is haplogroup G, the actual populations were more complex than a single genetic line. Associate Professor Ezgi Altanışık noted that the genetic structure of the regional people carried traces from communities on both the eastern and western sides of the Fertile Crescent. The people of Upper Mesopotamia, the region that encompassed Göbekli Tepe and surrounding sites, weren't a pure population. They were a crossroads. They carried genetic material from the Levant to the south, from the Caucasus to the north, from the central Fertile Crescent, from surrounding regions. This made them genetically diverse while maintaining their fundamental identity. And notably, despite this diversity, it remained relatively stable. The genetic composition didn't change significantly for roughly 1,000 years, suggesting that the population maintained some degree of cultural cohesion and selective breeding practices, possibly involving kinship-related traditions that preserved genetic patterns across generations. What's particularly revealing is what happened to this genetic diversity as populations began to spread. Once haplogroup G populations started migrating outward, once they began establishing new settlements in the Mediterranean and Europe, genetic drift took over. Populations that split off and moved to new territories began to accumulate their own unique mutations, their own genetic variations. This created the subclades of haplogroup G2A that we see distributed across Europe today. G2A P15, G2A M406, and other sublines trace specific migration pathways and settlement patterns. By analyzing modern populations and comparing them to ancient DNA recovered from Neolithic burials across Europe, geneticists have been able to reconstruct migration routes with remarkable precision. Populations that started in Anatolia moved south and west into the Mediterranean, settled Cyprus and parts of Greece, crossed into southern Europe. Other populations moved through the Balkans into central and northern Europe. Some eventually reached as far as Scandinavia and the Atlantic coast. This expansion wasn't instantaneous. It unfolded over thousands of years. Archaeological evidence suggests that the primary phase of Neolithic expansion into Europe began around 8,500 BCE and continued for roughly 2,000 years. This wasn't a single migration event, but rather a wave of advance, a gradual movement of populations who carried agriculture with them. Archaeological sites show a clear pattern. As populations moved into new territories, they maintained recognizable cultural practices, similar pottery styles, similar settlement patterns. Genetic data corroborates this archaeological pattern perfectly. When researchers use mutation rates in DNA to calculate how long ago populations diverged, when they use the accumulation of unique mutations in isolated populations to estimate divergence times, the numbers align with radiocarbon dating from archaeological sites. The result is a coherent narrative written in both stone and genes, a story that archaeology alone could never have told with such precision. The convergence of these two independent lines of evidence, genetic data and archaeological data, is what gives researchers confidence that their reconstructions of ancient population movements are accurate. But the story doesn't end with the Neolithic expansion. Subsequent migrations complicated the picture. Around 6,000 years ago, a different population began moving out of the Pontic-Caspian steppe region north of the Black Sea. These were Indo-European speakers, carriers of different haplogroups, particularly haplogroup R1b and R1a. They spread across Europe in a different wave, overlaying the existing Neolithic populations, creating the complex genetic structure of modern Europeans. This is why modern Europeans carry a mixture of genetic signatures. We carry the markers of the initial Neolithic farmers, the descendants of Göbekli Tepe's builders. We also carry the markers of the later Indo-European expansions. We carry the signatures of subsequent migrations and population movements. We are, in essence, a palimpsest of human history. Our genome's a written record of every significant population movement of the past 12,000 years. What makes this story particularly compelling is that it connects contemporary humans to Göbekli Tepe in a direct biological sense. If you are of European descent, there's a substantial probability that your paternal ancestry traces back through the Neolithic expansion to populations in the Fertile Crescent and Anatolia. Your genetic code carries the inheritance of the people who built Göbekli Tepe. You are descended from the population that experienced that crucial transformation from mobile hunter-gathering to settled agriculture. This isn't metaphorical. This is literal. This is written in your DNA. But here's where the story becomes more complicated and, in some ways, more fascinating. The genetic evidence tells us who spread and when, but it doesn't tell us everything. Genetic data alone can't explain the cultural innovations that accompanied these migrations. Genetics shows us that Neolithic farmers expanded from the Near East into Europe, but it doesn't explain why Göbekli Tepe itself was eventually abandoned. Around 8,500 years ago, the site that had served as a gathering point, perhaps a ceremonial center for thousands of years, was deliberately buried. Archaeologists found evidence that the site was intentionally filled in, covered with soil and rock, abandoned. This wasn't accidental decay or gradual neglect. The filling appears deliberate, intentional, ritualistic. Why? The traditional explanation was that as populations began to settle more permanently, as agriculture became more important than ceremonial gathering, sites like Göbekli Tepe lost their function. Nomadic populations who had traveled to Göbekli Tepe for seasonal ceremonies now lived in permanent villages scattered across the landscape. The need for the central gathering place diminished. Perhaps the burial of the site was a ritualistic closure, a deliberate act of farewell to a sacred place that had outlived its purpose. dwellings, artifacts suggesting year-round occupation, or at least extended stays. This has forced researchers to reconsider the site's purpose and function. Perhaps Göbekli Tepe was never purely a temple. Perhaps it was a sacred city, a place where the religious and the mundane coexisted, where priests and practitioners maintained permanent structures while pilgrims visited for ceremonies. This reframing doesn't contradict the genetic evidence. It complicates it, enriches it, but doesn't invalidate it. The most recent excavations have also revealed human burials, and most remarkably a life-sized human statue discovered embedded horizontally within a stone wall, remarkably preserved despite its age of over 11,000 years. This isn't a crude fertility figure or simple ritual object. It's a carefully crafted human form, suggesting that the people of Göbekli Tepe possessed the skill, the artistic vision, and the desire to create representational human imagery at a time when such sophisticated sculpture was unknown anywhere else in the world. Meanwhile, at the nearby site of Karahan Tepe, researchers uncovered the first T-shaped pillar bearing a carved human face, a revolutionary find that transforms our understanding of how Neolithic peoples represented themselves in stone. These discoveries suggest that whoever built Göbekli Tepe was capable of sophisticated artistic representation, of creating commemorative objects, of investing emotional and spiritual significance into human imagery. This speaks to cognitive sophistication, to complex beliefs, to a level of abstraction in thinking that rivals any of the great civilizations that would emerge thousands of years later. These weren't primitive peoples in any meaningful sense of the word. They were artists, architects, engineers, and spiritual leaders. The genetic story and the archaeological story are now merging into something more complete. We're not just looking at a population that expanded outward and populated Europe. We're looking at a population that possessed sophisticated artistic expression, that created permanent sacred architecture, that organized elaborate ceremonies, that developed complex spiritual beliefs. And this population was part of the Neolithic transformation. They were the farmers. They were the people who domesticated crops and animals. They were the population that would eventually build the first cities, the first states, the first empires. The genetic signature of haplogroup G isn't just a marker of ancestry. It's a marker of that crucial moment in human history when our species transitioned from a nomadic existence to a settled one, when the foundations of civilization as we know it began to solidify. What's remarkable is how recently this transformation occurred in human terms. Anatomically, modern humans have existed for approximately 300,000 years. For most of that span, humans were hunter-gatherers. The transition to agriculture is less than 15,000 years old. The building of Göbekli Tepe, approximately 11,500 years ago, marks a point very close to the beginning of that transformation. It predates agriculture in Europe by thousands of years. It predates most settled agriculture outside the Near East by even longer. Göbekli Tepe represents humanity at a crucial threshold, at a moment when our species was beginning to imagine permanence, to invest labor in structures that would outlast their builders, to create religious and ceremonial systems complex enough to demand permanent infrastructure. The genetic evidence suggests that the people who made this leap, who took that crucial step toward civilization, carried specific genetic markers that we can still identify today. When you look at the distribution of haplogroup G2A across modern Europe and the Mediterranean, when you see its concentration in regions that received early Neolithic farmers, when you see how it's distributed in a pattern that matches the archaeological record of ceramic styles and settlement patterns, you're seeing the genetic footprint of that transformation. The people who made that leap were biologically successful. Their genes spread, their children inherited their innovations. Their descendants became the farmers and herders of the Neolithic, and eventually the builders of the Bronze Age civilizations. What the genetic evidence cannot tell us, what remains shrouded in mystery despite decades of genetic research, is the specific mechanism of cultural change. How did the idea of building Göbekli Tepe arise? What motivated the investment of thousands of person-hours in construction? What ceremonies took place at the site? What beliefs did the builders hold? What changed in the human mind that allowed the transition from nomadic existence to settled agriculture? These are questions that genetics alone cannot answer. They require archaeology, anthropology, and speculation grounded in what we know about human societies. But the genetic evidence does confirm that this change happened, that it spread, that it transformed the world. And it happened first in the region of Göbekli Tepe, among the carriers of haplogroup G, among the Anatolian hunter-gatherers who would become the farmers of the Neolithic. Today, when geneticists look at the genetic composition of modern populations, when they trace paternal lineages back through time, when they compare ancient DNA from Neolithic burials to modern populations, they find overwhelming evidence for the expansion of Neolithic farmers from the Near East into Europe. The genetic signal is clear, but the cultural implications are equally profound. Every European, every person of Mediterranean or Near Eastern descent, Everyone who can trace paternal ancestry back to Europe or the Mediterranean carries the genetic legacy of the people who built Göbekli Tepe. Their genes are in us. Their innovations are in us. We are the products of that transformation, that moment when humanity decided to stay in one place, to build, to create civilization. The genetic story is also a story about adaptation and survival. The Anatolian hunter-gatherers who carried haplogroup G successfully adapted to changing environmental conditions. Around 10,000 years ago, the Earth's climate was shifting. The last ice age was truly ending. Temperatures were warming. Weather patterns were becoming less predictable. Populations of megafauna that had sustained hunter-gatherers for millennia, massive animals like woolly mammoths and giant elk, were declining or disappearing. The environmental conditions that had supported pure hunting and gathering for hundreds of thousands of years were becoming less stable. This wasn't a catastrophic change, but rather a gradual shift toward a different climatic regime. The domestication of plants and animals was a response to this environmental pressure. It was a solution to a problem. Archaeological evidence suggests that the people living in the Fertile Crescent began intentionally cultivating wild wheat and barley, began managing wild sheep and goat populations, began creating the first agricultural systems at least 10,000 years ago. And the people who successfully implemented this solution, who made the transition to agriculture, gained a competitive advantage. Their populations grew faster. They could support higher densities of people in the same territory. They could create surplus food, which could support craftspeople, priests, and warriors who didn't produce food themselves. They could invest in monumental architecture and complex ceremonial systems. And genetically, this success is evident. Their genes spread far and wide, eventually populating an entire continent. But here's an important caveat. The genetic dominance of haplogroup G in Neolithic Europe didn't mean the complete elimination of local hunter-gatherer populations. Genetic analysis of early Neolithic populations in Europe shows evidence of a mixture of interbreeding between incoming Neolithic farmers and local hunter-gatherers. The process was more gradual than a complete replacement. In some regions, the transition was more rapid and complete. In others, local populations integrated into Neolithic society, and their genes mixed with those of the incoming farmers. This is evident in the genetic composition of modern Europeans, who carry genetic contributions from both Neolithic farmers and the earlier European hunter-gatherers. The genetic story is therefore a story of both expansion and integration, of cultural diffusion and gradual genetic change. The implications of this genetic evidence extend beyond academic archaeology. Understanding the genetic basis of the Neolithic expansion helps us understand modern population genetics, helps us comprehend why certain genetic markers are distributed the way they are, helps us trace the complex pattern of human migration and settlement that has led to the world as it exists today. It also provides a sobering reminder of how recent, in evolutionary terms, the transformation to settled civilization actually was. We are not ancient peoples, we are very recent ones. The transition from nomadic to settled existence, which created all the conditions for the development of complex societies, states, and eventually modernity, happened less than 15,000 years ago. That's approximately 500 human generations. It's nothing in evolutionary time. It's barely a moment in the span of human history. This realization should provoke thought about the nature of civilization itself. We often think of civilization as a natural state, as the inevitable outcome of human development. We forget that for the vast majority of our species' existence, we were nomadic. We were mobile. We didn't build permanent structures. We didn't create states or governments or complex hierarchies. And then, very suddenly, in evolutionary terms, in a specific place, among a specific population, that all changed. The people who built Göbekli Tepe made a choice, perhaps not consciously, perhaps driven by environmental pressures and cultural innovations, but a choice nonetheless. They chose to stay. They chose to build. They chose to create something permanent. And that choice, written in their genes, spread across the world and shaped the destiny of the human species.